Hello students, welcome back to KapMan Academy, and today we are going to talk about an example of nitrogen fixation which is done by the bacteria Rhizobia. Right, so this nitrogen fixation, it is a very good example of symbiotic relationship between plants and bacteria. Plants and bacteria. And in this mechanism, what happens? Again, that atmospheric nitrogen, it is converted into the organic ammonia form, right? And this process here, it is called nitrogen fixation. And this process, it is very much useful in the agriculture because it actually enhances the quality of the soil in the field. And the nitrogen fixation where it occurs in the plants, those plants are called your legumes plants. Those are your legumes plants. Now if we talk about the bacteria which is actually responsible for the nitrogen fixation, so that one is called Rhizobium bacteria and its relatives, which are alpha proteobacterium, which are known as alpha proteobacterium and beta proteobacterium. If we talk about their examples, then in case of alpha proteobacterium, there are Rhizobium, right? There are Rhizobium, there is Rhizobium, and there is Rhizobium. So they all are relatives of the Rhizobium bacterium, and they also do the nitrogen fixation. If we talk about the beta proteobacteria, in that case the examples are your Burkholderia caribensis, Burkholderia caribensis. It's the example of beta proteobacteria. Another one is your Ralstonia taiwanensis. Right? So they are also responsible for the nitrogen fixation. So they all together, Rhizobium, alpha proteobacteria and beta proteobacteria, they all together known as the name which is called Rhizobia. And if we talk about the property of these Rhizobia, so they are free living bacteria. They are free living bacteria present in soil, right? And these bacteria, they actually invade the plant roots. They actually invade the plant roots. And for the plant, they act as an invader, or you can say as a alien invader. So since they are the alien invader here, so plants actually, they act against these bacteria. So in response to these bacteria, plant actually produces oxidative burst. And this oxidative burst, as we know, it contains different type of superoxides and its intermediate hydrogen peroxides and different type of NOI, right? So Rhizobacteria, it acts here very smartly because here the plant is actually producing the oxidative burst to kill the Rhizobium bacteria. But Rhizobium bacteria act here very smartly and survive. How it survive? Because Rhizobium, it actually contains antioxidants, and these antioxidants, they help the Rhizobium to actually continues its infection in the plants. It continues its infection in plants. So this is how rhizobia actually bypasses the plant defense system, right? So when the rhizobium actually survive in the plant defense system, then finally the plant roots, they actually induces a inducer molecule. They actually produces, the plant finally, it actually produces a inducer molecule in response to the rhizobium, right? And this inducer molecule is known as flavonoid inducer molecule, right? And this flavonoid inducer molecule, it actually induces the rhizobial rhizobial colonization colonization in the plant roots. So when this inducer is actually being produced from the plants, then further rhizobia also produces a signaling molecule. Rhizobia also produces its signaling molecule, and that signaling molecule is known as nod factors. And these node factors are actually responsible for the formation of nodules in the plant roots. Plant roots, right? So as you can see here in the image, you will be clear that what is happening. So in this case, what is happening, that your rhizobium actually, here we are talking about, so in the nitrogen fixation, your rhizobia here, it is actually bypassing the plant defense system. How? By actually producing the antioxidants against the oxidative burst, and then further plant actually, it produces the flavonoid inducer molecule, and that flavonoid inducer molecule induces the production of signaling molecule from the rhizobia, which is known as nod factors. Now we are going to talk about these nod factors, how they are actually responsible for the formation of nodules, right? So if we talk about the nod factors, then they are your molecules which are consist of 4 to 5 units of beta 1 beta 1 4 linked N acetyl N acetyl D-glucosamine, N-acetyl D-glucosamine, right? And they actually enhances the gene expression in the outer epidermal cell layers of the roots. So if it is your roots, then they actually enhances the epidermal cell layers gene expression. Gene expression. Because of what? Because of the production of node factors, right? Now, due to the production of node factors, the root here, it starts to get deformed, right? And it actually takes the shape which is known as Shepherd's crook, as you can see here, right? So this Shepherd crook is basically to entrap the rhizobium bacteria in the root hairs, and they actually forms a infection thread. So as you can see here, this is the formation of infection thread in the root hairs, right? Upon formation of the Shepherd's crook, what happens, that this infection thread actually, it starts to grows towards the primordium of the root hair, this region, primordium region of the root hair, and here it finally forms the nodule like structure, nodule like structure finally, right? Or you can say a tube like structure. So this Shepherd crook is to entrap the rhizobium, and this actually the tube like structure here, it is an infection thread, and together they all form the nodule structure, right? So upon formation of the infection thread, further the rhizobia actually, it is gets released in the root hairs, right? And it gets released as because it is protected by the plasma membrane of the plasma membrane of plant, right? As you can see here, inside here it is the rhizobia bacterium, and outside here it is the plasma membrane of the plant, and together they are known as bacteroids. Bacteroids. So as you can see here, now as rhizobia release, it actually starts to divide, right? And as it divides, it takes a structure which is known as symbiosome, and this symbiosome are your finally nitrogen fixing sites. Why the symbiosomes are actually being formed? Because as we know, for the nitrogen fixation we need nitrogenase enzyme, and we already had mentioned nitrogenase is your oxygen sensitive enzyme. So to do the nitrogen fixation, it has to be somewhere in the enclosed cells. So here are the sites for the nitrogen fixation where there is no oxygen around the nitrogenase enzyme, right? So here the nitrogenase is protected by the symbiosome structures, or you can say the nodule structures, right? Other than this, nitrogen is also protected by a leg hemoglobin molecule. It is also nitrogenase, it is also protected by the leg hemoglobin molecule. How? Because leg hemoglobin, it actually binds to the oxygen, and it actually makes the oxygen unavailable for the nitrogenase so that it cannot get oxidized further, right? Apart from that, symbiomes, as I said, they actually are the nitrogen fixing sites. So here they actually convert the nitrogen, atmospheric nitrogen, into the ammonia form, right? And in the return, these microbes, they actually provides the amino acids, newly formed amino acids, to the plants, and in the return they take carbon and energy for their survival, right? So for the nitrogen fixation in the rhizobia, here they actually need some essential genes. They need some essential genes. If we talk about the nodulation, then they are nod factors such as nod, NOL genes and NOE genes. Their expression is important for the nodule formation. If we talk about the nitrogen fixation, then we need NIF genes and we need fix genes for the nitrogen fixation, right? And all these genes are actually present in the Rhizobium bacteria, and their location on the genome, it also varies from one bacteria to another bacteria. So this is all the story of nitrogen fixation in the leguminous plants by the Rhizobium bacteria. So if you see in this image, which is given above here, now it will be very clear to you how the nitrogen fixation is occurring in the leguminous plants, right? So first of all, here there is a presence of nodule in the root hairs, right? Formation of the nodule, and it is occurring because of the nod factor activation, right? As the nod factor gene activates, they start producing a nod factor which initiates the nodule formation. Nodule formation actually makes the deformed root hair which curled towards the root primordium region and forms the chamfered crook shape here, right? Where they starts to divide in the form of bacteroids and starts to fix the nitrogen. So this is all the story of nitrogen fixation in the leguminous plants by the Rhizobium bacteria, right? So that's all for the today. Thank you.